My name is Jacob, and over the past 3 years I have been working with Dr. Misty Paig-Tran to understand the morphology and filtration efficiencies of whale shark filter pads. Rhincodon typus is established as the largest fish in the ocean and can measure up to 18 meters in total length. This species can be found on either side of the Pacific Ocean, South Africa, and the Indian Ocean. In addition to its size, it is also known for being one of 3 filter-feeding sharks alongside the megamouth shark and the basking shark. It does so by 3 behavioral feeding mechanisms: subsurface passive filter feeding, surface ram filter feeding, and stationary suction feeding. During feeding, it typically swims at slower speeds and filters out particles microns large. How it goes about this is proposed to be a form of biological cross-flow filtration. In this form of filtration, fluids flow almost parallel to the filtering apparatus and either get caught in mucus or get knocked back into the mainstream flow. Although much work has been done in identifying cross-flow filtration in suspension feeding fishes, little is confirmed about the mechanisms for whale sharks. One of the main issues as to why this is so poorly understood is that getting a hold of adult specimens like this one are difficult. Geographically, the nearest distribution range of whale sharks end off the coast of Baja California. And the difficulty of capturing these organisms for study comes from being established as an endangered species back in 2016 and is protected by countries like the US. Where individuals are caught commercially is geographically inaccessible, like Pakistan or Taiwan. Thanks to a study by Dr. Paig-Tran in 2011 with her colleagues, reassessment on how we go about studying these organisms may change. The use of preserved neonatal specimens and constructing physical models of these organism structures enables us to systematically test their unique physiological processes. For this study, because neonatals have been established as proxies for adults by Dr. Paig-Tran and her colleagues, we want to determine how our neonatal specimen compares to filtration morphologies in adults. And from these morphological measurements, we want to develop a biologically relevant model to understand filtration properties of the filtering apparatus, the filter pads. Here we have an adult whale shark from the Gulf of Mexico next to the bucopharyngeal cavity of our neonatal whale shark specimen. The adult whale sharks that we are comparing to are those described by Dr. Moda and his colleagues in their 2010 study. On average, the total length of the neonatal is 10% of the adult total length. The bucopharyngeal cavity and mouth are both elliptical in nature, with adults having over 100 times the mouth area as neonatals. The 5 distinct dorsal and ventral filters on both sides of the pharynx create a total filter area in the neonatals that is 1% the reported area in adults. The total of 20 filter pads are connected on their posterior edges by a combination of the epibranchial and ceratobranchial arches, with rafts of connective tissues providing flexibility to the cavity. For both maturities, the first 4 filter pad sets are roughly rectangular in nature, with the 5th set geometrically resembling more of a triangle or a trapezoid. Looking more closely at the filter pads, we see strong comparisons in the filtering structures. Here we have an adult on the left and the neonatal on the right. Both are composed of hyaline cartilage, and neither hosts goblet cells for the production of mucus. The identifiable structures are the primary filter lobes, secondary filter lobes, or gill rakers, filtering gaps, and pores. Dermal denticles can be seen on the adult and be developing on the neonatal, shown here. The reported porosity ratios for adults are on average 53%. Using photo editing to increase the contrast of the open area to the biological structures and MATLAB image processing, we see that our filter pad areas are roughly 97% open area. The neonatal average pore size is 16% of the adult pore size. This is interesting, given that the adult filter lobes are on average 10 times the thickness as the neonatals. The increased pore diameter could be contributed to expansion during development, since the adult filter pad is 8 times the width of the neonatal. When looking at the distal end of the filter pad from the parabranchial chamber, we see both the adult and neonatal have developed primary and secondary veins that provide structural support of the gill rakers. Notice that the primary veins in the adult filter pad begin to come together while the neonatals remain distinct. The crowning primary veins and the increased filter lobe thicknesses may reduce the filtering gaps and thus account for the reduced porosity. In order to complete our understanding of the filtration conditions, we must understand the tangential and transverse flows. Here we see the ventral filter pads proximal to the parabranchial chambers. Tangential flow refers to the main stream that introduces particles to the filtering areas. Transverse flow refers to fluids that pass through the filtering apparatus. By sectioning the bucopharyngeal cavity into 6 distinct stages and using the continuity equation, we can find the tangential flow velocities that will interact with each filter pad set. We then convert the cross sections into circular pipe diameters using the hydraulic radius of an ellipse, which allows us to calculate the average Reynolds number throughout the bucopharyngeal cavity. This is a dimensionless descriptor of the difference between viscous and inviscid flow. The higher the Reynolds number, the more turbulent the conditions are. In our system, the average tangential Reynolds number is about 15,000. This can be used in our models since functionally there is very little difference between Reynolds of 17,000 versus 13,000. When assessing the transverse flow, we combine the results of our porosity with our geometric model and use similar calculations to the tangential flow. In doing so, we come out with a Reynolds number for the entire filtering area as about 17,000. 5,000 for a single filter pad, and 9 for a single pore. To account for their prey items, we saw that within their feeding areas, the most common prey items available are surgisted shrimp, chaetognathes, copepods, and fish eggs, listed in order by their abundance with average maximum sizes below. This may fluctuate though, since stomach content analysis shows a much wider range of available prey sizes for copepods and fish eggs. Since most of the stomach contents are mostly composed of surgisted shrimp and chaetognathes, if size selectivity does exist for their filtration, we may see selection for larger particle sizes. In the final stage of the model, we create the physical filter by hand tracing a dissecting microscope image of the filter morphologies and converting it into a 3D model. I then sent it to our Formlabs 3D printer for construction. And after the design was printed, it was washed, cured, and finally ready for use. The design is meant to be fit to an acrylic pipe that serves as our model fish. We then use a constructed crossflow filtration apparatus to control the flow rate that interacts with the filter, inject particles, bring it to a steady state, and simulate consumption by having 2 outfall sites, the filter and the simulated esophagus. Particles ranging from 125 to 425 microns were captured using a 100 micrometer mesh over the course of a minute. Any remaining particles after that minute were flushed out of the system in weighed. At a Reynolds number of 27,000, which closely resembles the transverse flow through filter pad set 1, we see that the proportion of the particles that are captured in the simulated esophagus increases with decreasing particle size. These pie charts represent the average particles collected after each trial. The blue represents the particles that were captured at the filter outfall site. the orange for the particles captured at the esophagus, and the gray for the particles that did not participate in filtration. Efficiency was used to assess the amount of particles captured by the esophagus that participated in the filtration. The negative values indicated that there was more mass collected than what the experiment started with. This mass could be attributed to external contamination of dirt or the variable wet weight. In this study, we established morphological comparisons between our neonatal whale shark with adults, documented in primary literature. Some of the results to highlight show that the development of dermal denticles may imply protection from incoming food particles and reduce drag on the surfaces. This closely resembles ricochet separation filtration mechanisms described by Devi et al. In 2018. For the flow conditions, larger Reynolds numbers have been shown to increase size selectivity for smaller particle sizes, which is what we see here in our 27,000 Reynolds number flow conditions. For our future studies, we want to reset the flow conditions to a tangential flow of about 15,000 and add a backflow apparatus to create a transverse Reynolds number of about 5,000, which will provide more biologically relevant conditions. For the filter, I would like to reevaluate the porosity to confirm our findings, since the denticles could have been mistakenly counted as empty space during image processing. I also want to include primary and secondary veins to the filter. In addition to repeating these size ranges, the last thing to do would to increase the range to finish establishing the size selectivity gradient. With that, I would like to thank the Fab Lab for their continued support throughout this process. Raj Devi was the graduate student in the lab who helped me get my bearings and design the PVC apparatus that I use for my filters. Dr. Paig-Tran has been very supportive and patient throughout this process, giving me ample opportunity to learn and grow, while also making sure I stay grounded with my ideas. Dr. Philip A. Hastings and Dr. Ben Frabel at the Scripps Institution of Oceanography should be recognized for their support of this project by lending the study specimen that was used. The Southern California Ecosystems Research Program, with their associated donors, Dr. Mike Horn, Dan Black Gift Challenge for Undergraduate Research, the BEIM Foundation, and the Council on Oceanic Affairs, Science, and Technology, were all valuable funding sources to many of the materials. SERP also helped develop various research and professional skills that I will be sure to use to the end of my career. Dr. Ed Reed was very gracious in providing space, electricity, and water needed to run the apparatus at the greenhouse. And last but not least, I would like to thank Dr. Steve Carl for assisting me with the microscopic imaging of this filter pads. Thank you.